Eric, are you ready?
Thank you.
Yeah, I'm ready.
All right. So it's my pleasure to introduce Dr. Eric Potma. He's a professor of chemistry and professor. Also, he's got an associate position, affiliated position with biomedical engineering. I think with electrical engineering as well. And he's a faculty member of the Beckman Laser Institute. He is a guru in nonlinear optical microscopy. He's a pioneer in coherent anti-Raman Stokes microscopy. And I'll let him do the rest of the introduction for himself, but he's, three-dimensional Eric would have been fantastic to have in this room. He's a very dynamic speaker, hopefully conveys in 2 dimensions.
And tall.
So, Dutch.
All right, folks. Thank you, Bernard. Really appreciate that introduction. Yeah, I would have loved to be there, and I kind of am because I'm actually in the same building. I just sneaked in this morning very early. I'm in my office. I actually am kind of fully recovered from COVID, so kind of tested negative, which is great. I just don't want to, take any chances with you guys, so that's why I'm just playing it very safe. So, any case, I will do it online like this. Hopefully it works out. And we're going to talk about, let's see, this story, right?
Plugging his book there too. I don't know if you saw it. Yes. He has a fairly new book. He wrote the whole thing himself.
Oh. You advertised my book, Bernard? Thank you.
Yeah, it was on your table, so I said I thought I should mention it.
All right. Okay, so we're going to talk about optical microscopy in cells and tissues, and my expertise is really in nonlinear optics, nonlinear optical microscopy. So we will definitely focus on that mostly. But I just want to start with from very simple principles, humble beginnings, and then we'll just touch on some additional more advanced topics. So. What you will be hearing is maybe some of you already know this and you feel like, I learned nothing new. I'm sorry. But I think, just by presenting this presentation, hopefully you will take something away from it. The emphasis is really on, what can you extract from, what information can you extract from the sample using these kind of nonlinear optical techniques, and when is it not a good idea? So that's kind of like the main scope. Microscopy is fantastic. It's a great way to investigate biological samples, and the key is really to have a lens system that has a proper magnification and resolution. So these objective lenses are fantastic. I mean, nowadays they are just approaching their theoretical limits, and you purchase one for a couple of $1,000s, and you can see things at the submicron scale, which is just absolutely brilliant. And there's 2 ways to do it. The simplest way to do it is just to illuminate your sample with a random light source, just a lamp that produces light in no particular direction. And you basically put it through the objective, you illuminate the sample, and then through the same objective, you can just see if there's any light coming back. That can be fluorescent light. You use a couple of filters, and then you project it onto a camera. So that will be bright field or wide field imaging, and you get beautiful images out of that. But I will not be talking about that kind of microscopy, because I will be talking about microscopy where we use laser beams, which, of course, as you know, have very distinct properties. They have collimated light, so the photons in those beams, they have a spatial phase relative to one another, which means it propagates in a beautiful beam-like fashion. And those beams we can focus really well to small spots. And these small spots are submicron in size in most cases. And that means that we can scan that spot across the sample very rapidly. There's all kinds of ways to do that. We won't go into the details. But the principle is that from that spot, we collect the light that's coming back, project it back onto a very sensitive detector that doesn't have any spatial resolving capabilities itself, but it can detect the light from one spot, and we time synchronize our signal with the scanning optics, and then we reconstruct our image in the computer. So this is laser scanning microscopy. So it requires you to scan the laser spot in the sample, and then you can take advantage of the fact that the detector is super sensitive kind of a single pixel super sensitive detector, like a photomultiplier or a kind of a similar type of detector. And that is kind of the kind of microscopy that we're going to talk about. So this type of microscopy makes use of highly focused laser beams. And you probably know what those laser beams look like when you focus, but there's some interesting stuff happening here, because if you look at this focal volume, and what you see here is the focal volume projected along the so-called ZY plane. The Z-axis is the propagation axis, and the Y-axis is an axis orthogonal to that in the lateral direction. And so you have maybe seen those kind of cigar-shaped focal spots, and you're not too surprised about it, but it's still interesting because this focal spot, of course, is the result of all these waves coming together and constructively interfering. In the middle, that is your focal spot, and next to it, you see some dark spots because there's destructive interference happening. And so this is a diffraction effect, right? So the fact that we look at these little tiny spots is a result of light diffraction. And the fact that we cannot make a little spot that is circular or spherical in all dimensions is because we cannot have light beams coming from all directions at the same time. We only illuminate it from certain angles, and therefore we are imperfect along the lateral or the axial coordinate. And therefore along the axial coordinate, we have an elongation going on. Okay, so, and that's something we can perfectly fine live with. The resolution that we can get from this focused light is sufficient for most applications. And therefore, it's just a convenient tool for optical microscopy. So even though you may be familiar with this picture, you may not be familiar with the next picture, which is not the amplitude of the field in the focus, but actually the phase of it, or the relative phase of the optical field as we move through focus. And what you see here is kind of a phase map, okay? So we go from hot to cold. That is basically a 2 pi phase shift, or a pi phase shift, I'm sorry. And you can see that there's a phase swing across the focus. If you look here below, that is like this the hot color, and if you look on the other side, above focus, you see that it is blue, which means we have undergone a kind of a pi phase shift. Okay, so this is a well-known effect. It's called the Gouy phase shift, and this actually has no real effect if you do fluorescence microscopy, but it does have a real effect if you do other forms of microscopy, such as coherent nonlinear optical microscopy. This phasing is actually really important. And so we'll be talking about this a little later on in order to see if we can understand it. Okay, so the width of the focus, roughly about 300 nanometers, is very typical. And the depth of focus, which is basically the length of this cigar, is approximately a micron or so, depending, of course, on your color of light, the kind of the numerical aperture of your objective lens. But those are kind of very typical numbers with which we can do the biological imaging. So if you look at this, kind of an additional rendition of this focus, now in kind of an hourglass form, because we want to indicate that light really comes from below and then travels through focus. The light gets condensed, and then it kind of wavers out again, okay? And we are drawing a line here in the middle. That is our focal plane right there, okay? And we're going to integrate that light here, and I call it S. So that is just the light intensity in focus, all right? And as you can see, in the middle here, that's my focal volume, okay? But the question that I'm asking myself, what happens if I lower this detection plane? So what if I now move it kind of beyond the length of the cigar? Okay, so let's say here. So this plane right here, what is the intensity of the light in this plane? Okay, who knows what the answer is? Anybody?
Sorry, I was writing something on the board. They might have been watching me. I don't want to interrupt you, but I was basically saying if anyone has a question, they need to speak up because I don't think you'll be able to see them.
Okay, very good.
So you might have to say this again.
Okay, I'm going to say it again. Who knows what is the intensity in this plane below it? I mean, basically, the question is it lower in intensity, higher, or maybe the same as the intensity of the plane over here?
It's 0.2 times lower than the central intensity.
2 times lower, I hear.
Sorry, root 2 times, not 2 times.
About 2 times.
Root 2 times.
Root two.
Square root.
Square
Square.
root, somebody says. Okay. All right. Well, that's a good guess, but it's not correct. Okay. I mean, what I'm doing here is I'm integrating the light in this entire plane. It still adds. Why is that? Because the light basically has to travel from below through the middle. It doesn't matter if you compress it a little bit. The total is still the same. It's just a light flux through the whole thing. Okay. And you're like, okay, I get that. That's actually very important. That's very important because if you do regular microscopy, the fact that you waver out a little bit, but the same number of photons still has to travel through such a plane, and therefore those planes are still being, the molecules in those planes are still being excited, and they can still contribute to light, right? So all these planes, they all basically have the same photon flux through them, and they can all produce optical signals that you will be able to detect, okay? So the thing is that there is no real depth dependence in terms of the total intensity. The total intensity in regular light microscopy is not optically sectioned, so to speak, okay? Because every plane contributes in terms of photon flux the same, okay? So if you do regular imaging, let's say you integrate it onto a camera, then all these planes will somehow contribute to the total signal. Now, not all those planes will be in focus, so the planes below will contribute something that is out of focus. That is true, but they still contribute light, okay? So this is, of course, the effect. If you do something that can reject all that light and only see your focal plane, you see, of course, a very bright image. This is a fluorescence image. But if you do the same thing and you don't reject that light some way, somehow, then you see the image on the right. So, and that is because all those planes contribute to your signal. So that is something that people, of course, don't like, and they have come up with a whole bunch of ways to reject that light. And the main solution is you have to kind of find a way to reject the out of focus light. And there's all kinds of ways to do it. The main thing is confocal detection. That is a technique invented in the 1960s, I believe. And what you want to do, you want to just select only those photons that come from this cigar here and reject everything else. And the way you can do that is just send everything through a pinhole. That's a conjugate of this cigar in a way in front of your detector. Okay, and what happens is you are only sensitive to light that emerges from that region right here, but it's really based on directionality and kind of the diffraction properties of unperturbed beams. So there's a kind of a connotation there. Okay, but in principle, confocal detection works beautifully well. It quite effectively rejects out of focus light, and therefore you retain again the information that only comes from the central portion of your focal field. Another way to do it is to do it not on the detection side, but actually do it on the excitation side. So let's not excite anything out of focus in the first place. And that is what nonlinear excitation allows you to do. In nonlinear excitation, we take advantage of the fact that there's a nonlinear dependence of the optical signal with the intensity of the incoming light. And that is definitely much higher in the cigar than outside of the cigar. It kind of tails off non-linearly. And so we are suppressing much more the out of focus contributions than we do for the in-focus contributions. And so that is a beautiful way to do the same thing. And in fact, nonlinear excitation has some clear advantages over confocal detection. So nonlinear optical microscopy is indeed based on this very simple principle. I have an optical signal that scales in a nonlinear fashion with the intensity of the incoming light. Okay, so that is a very simple definition, but it's important to understand that it's the intensity to a power n, where n is an integer larger than 1. That is simply the definition of an optical nonlinearity. Okay, and there's all kinds of ways you can do that. You have, for instance, a signal that depends just the square of the incoming light, but you can also have something that depends on 2 light beams coming in. Let's say I have 2 different colors, color 1, color 2, and it depends linearly on both, but together, non-linearly on the total. Okay, so that is another way to create a nonlinearity. Yeah, if you guys have any questions, then please raise your hand or something so I can see what's going on. I have a little video of your room, but I cannot really see it. It's very tiny. Okay, so this is the same kind of plane scan that we did in drawing the plane across focus, but now I'm going to just plot it as a function of the axial position. So I'm going to start at a minus value and move through focus to a positive value. Okay, the u here is a unitless quantity that runs from, in this case, runs from minus 30 to 30. And what you see here is like three different kinds of optical responses. One is a linear response, that's the blue. And you can see, as we have seen before, if you integrate in that plane, then it is not changing. It is just completely straight. That means there is no dependence here on the depth for a one photon process, okay, when it's linearly dependent on the intensity. Now, if you do a two photon process, like two photon absorption, for instance, 2 PA, two photon absorption, then we're looking here at this curve, this orangey curve. It goes up, peaks here, and then it kind of falls down again. So that is clearly a case of optical sectioning, because if you move away from focus, there the effect is very little, okay? And the same is true for a three photon excitation process, let's say three photon absorption, 3 PA, that is indicated in red. As you can see, it's a little wider here for the 3 PA because I'm using a longer wavelength in this case. But importantly, and we'll get back to this one here later on, is that the tails are decaying much steeper, okay? So you see, compared to two photon absorption, the three photon absorption tails off way quicker. And that is really significant if you want to do deep tissue imaging. Okay, so this plane scan kind of example, very simple, but it highlights a very important characteristic, and that is this optical sectioning capability. The higher the nonlinearity, the better your sectioning capability. If you want to reduce out of focus light from the excitation side, the higher the nonlinearity, the better off you are. Okay, and this is an example that everybody shows that talks about nonlinear optical microscopy. This is the image taken in the late 90s. It's just a beautiful illustration of the power of nonlinear optical microscopy, because you can see in the linear version, which is the top of this, what happens here in the cuvette, you see this whole cone of light is glowing up. This is a fluorescent solution. It's excited with one photon excitation, and this whole cone is bright. But in the case of two photon, you only see this little tiny spot here. So the out of focus regions simply don't contribute to the signal. The molecules are not excited. And that is, of course, very attractive for biological imaging. Okay, so here's a couple of examples, of some random examples, really. This is confocal microscopy. You can do beautiful imaging. Out of focus light is rejected, multiple colors, but you can do the same thing in two photon. And the nice thing is that, if you do it in two photon, you take advantage of the fact that your wavelength is twice as long, and longer wavelengths have less scattering, and that means you can go deeper, okay? You can go way deeper into the tissue, and that is, of course, very attractive for biological imaging. And that is one of the reasons, or mostly the reasons, why two photon microscopy or three photon microscopy has become superior for tissue imaging applications. Okay, so if you do tissue imaging applications, live tissues and so forth, really nobody is using confocal microscopy. It's all nonlinear microscopy based because of these clear advantages. Okay, so this is a graph. I don't know if you have already seen this one in this short course. Maybe you have, but it is very important graph, okay? Because it shows on the y-axis the attenuation in reciprocal centimeters of the light in the tissue, okay? So the higher we go up on this scale, the more attenuation of light, okay? And on the x-axis, we have the wavelength of light. And there's a lot of information in here, although it's definitely not complete, but it indicates the attenuation caused by several mechanisms and different components of tissues, okay? So let me just highlight a couple of them. The first one is this kind of magenta line, which is the scattering contribution, okay? So as you can see, as expected, scattering is much higher at shorter wavelengths, and then it kind of comes down, all right? Kind of comes down, keeps going down. This is a logarithmic scale. So scattering generally, of course, is much lower for longer wavelengths. So that's a good reason to choose longer wavelengths. All the other curves are actually absorption curves, so attenuation caused by absorption of light. And one of the things I would like to point out is the green curve. That is not scattering, but that's actually absorption of melanin, okay? If you have melanin in your tissue, then that is a major contributor to absorption of light and therefore attenuation of light. Now, the wiggly curves are absorption spectra of different components, different from melanin. And here on the left, we see blood. Blood has, of course, a couple of well-known peaks here. And there's also a difference between oxidized and deoxidized, okay? So deoxy and oxy blood have different kind of absorption spectra. And then here on the, so this was on the left here, and on the right, we see two curves. One of them is water, that's the black one. It has a couple of peaks. These are actually vibrational overtones, okay, that extend all the way to the visible. And then this one here in blue, that is the absorption of fat, okay, in general. So fat as an overall class of materials. So if you now look at this whole graph, what you want to do, you would like to reduce the absorption and scattering at the same time because both contribute to attenuation. If you look at the area like right here, this area, you know, around 1,000 nanometers or so, you see that the overall absorption is pretty low because blood is absorbing very little. Also, you have very little absorption from water and fat. And at the same time, scattering is kind of pretty low, okay, already. So that is a very good range to be in, okay? So that is actually one of the reasons people like to be there. Now, if you are moving to the next so-called window, that window is chosen based on where water has its dips in the absorption spectrum. So the first window is about here, just beyond like 1,000 nanometers or so. You can go even further down, okay, but then scattering takes over. The second window is where this dip happens over here in the water absorption spectrum. That's 1,300 nanometers. And then you can go out even further. Let's say the dip over here, around 1,700 or so, is also attractive. It's attractive mostly because scattering is extremely low there, so you can go very deep. And water is really your limiting contributor here. Okay, so this graph is really useful in choosing which wavelength to use. Now, the nice thing about nonlinear optics is that we can use these very long wavelengths, use an optical nonlinearity, and then effectively excite chromophores that are still having their absorption in the visible range of the spectrum. Okay? So that is therefore a very attractive strategy. Okay, so scattering is, of course, a very important effect, and it affects optical microscopy in numerous ways, okay? So this is just a kind of very pictorial illustration of some of the effects that are happening. If I'm trying to focus my light in the tissue, I try to create this perfect kind of cone of light with this beautiful cigar that is, you know, maximally compressed in space. But what happens, of course, is that light scattering will try to counteract that, okay? It counteracts with this perfect diffraction situation, change the direction of photons and so forth, and therefore the diffraction that makes this cigar very tight in space becomes not so tight in space anymore, okay? So what happens is the focal volume becomes aberrated. And at the same time, if this light from this focus that is now a little bit larger is starting to radiate, that light may also be redirected by scattering, okay? So those are problems that we have to deal with if you want to do microscopy in tissues and cells. So if you do regular confocal microscopy, you use typically light in the visible that is like to the blue side, okay? So in confocal microscopy, of course, the goal is to excite molecular chromophores. A lot of molecular chromophores have their absorption spectra around 400, 500 nanometers or so. And that light is pretty blue, and therefore the primary contributor to loss of image quality is actually optical scattering, okay? So the focus, therefore, will be distorted, and that immediately leads to an elongation and a broadening of the focal volume. So the resolution goes down. The resolution goes down, the amplitude of the light also goes down, so you get less signal back. At the same time, the photons that come out are redirected, so you also lose those. Okay, if you use confocal, you cannot see them. They're gone. They will not go through your pinhole anymore. And so you see an overall reduction of the light flux. So even though you have this confocal pinhole that gets rid of all the out of focus light, you know, if all the light that is basically generated is going in different directions because of scattering, nothing is going to go through your pinhole anymore. So the signal is really steeply decreasing. So if you do nonlinear optical microscopy, you know, the same thing happens on the excitation side, okay? Your focus is still distorted. But the nice thing is about nonlinear optical microscopy that you don't need this confocal pinhole. So even though the photons that come out may still be kind of like diffusively coming out of your sample, they don't have to go through a pinhole afterward. You can just use kind of a wide area single pixel detector and try to capture them all, right? And therefore, you can actually get quite a bit of light because you don't care about the directionality of the light after the signal was generated. You just want to capture it. You just want to know how many photons were generated. You don't care about the directionality. So that's why nonlinear optical microscopy is really attractive for tissue imaging. Okay, so a little table here. Why would you like to do NLO microscopy? So the nice thing is that if you do nonlinear excitations, you can actually address a whole bunch of endogenous compounds in tissues and cells. So you get label-free imaging in principle. We'll get down to that in a moment. In linear microscopy, you can do the same thing, but usually linear microscopy is synonymous with you know, using labels. And using labels, of course, is a really, really good strategy. And most cell biology structures studies are based on using fluorescent labels. But in tissues, you cannot always start labeling things, especially in vivo and so forth. Labeling is much harder. So if you can derive contrast in a label-free fashion, then you're much better off. So penetration depth is much better in nonlinear optical microscopy because we're using longer wavelengths. And it is the opposite, of course, in linear microscopy. You use very shorter wavelengths, and that means more scattering. That means less signal. The resolution is intrinsically three-dimensional because the excitation is confined in space, and therefore you will not need a pinhole, and therefore you get more signal. Okay, so that's really attractive. You can use a wide aperture detector. You don't need to use the pinhole. An interesting thing is that the photo damaging effect, because you will always have to contend with photo damaging in any kind of experiment, is more or less confined to the focal range, okay, the focal region, because we don't excite molecules outside of that region. Now, if you do a linear microscopy, you don't really think about this, but basically since the light flux is the same all the way through, molecules get excited all the way through, and therefore molecules can get damaged all the way through. So they get damaging through the entire light cone, not just the focal region, okay? So that's why most of the time, the photo damaging in nonlinear optical microscopy is just confined to the focus, and that is also attractive. But, and of course there's a big but, the photo damaging in focus can be quite significant, okay? Because there, optical nonlinearities are amplified, not just your favorite optical nonlinearity, the one that you're interested in for your experiment, but also other nonlinearities, ones that lead to photo damage, okay? So you have to be very careful about how much light you send in. That is not the case in linear microscopy. So usually the light fluxes are less, the density is less in focus actually than it is for nonlinear in terms of the damaging potential. Also, it's actually quite expensive to do a nonlinear optical microscopy because you need special light sources, while in linear microscopy you can use much less expensive light source. And this is sometimes very important because not everybody can afford expensive machines, and therefore it's just less you know, applicable and accessible to everybody else. Okay, so let's say you have an optical microscope like the one in the middle, and you do have the funds to buy these special light sources. You can now do nonlinear optical microscopy, but you can do all kinds of nonlinear effects in focus, right? And here's just a couple of examples. I'm going to walk you through it very quick. Here on top, you see the coherent anti-Stokes Raman example. I'll highlight that a little later. This is its cousin, stimulated Raman scattering. Both of these are sensitive to molecular vibrations. Some frequency generation, that's this kind of Jablonski diagram here. This is actually going to be demonstrated in the lab tour that will follow after this lecture. Okay, so some frequency generation. One of my students will be showing you the setup, and she will be showing you how you can use this technique to image collagen. Second harmonic generation, also a great technique to do collagen imaging. You may have heard of it. Two-photon excited fluorescence, of course, the main workhorse, very powerful. And then pump probe microscopy, maybe you have not heard of this, but also very powerful way to actually see molecules that absorb, really hard to see otherwise because they don't fluoresce. They absorb a lot, they don't fluoresce a lot, so you can't see them in fluorescence microscopy. You can see them in pump probe microscopy quite efficiently. So just an example of things you can do if you have such a microscope. All right, are there any questions so far?
I don't see any.
All right, very good. All right, so then what I'm going to do is I'm going to just dive in a little bit into what happens if we are driving these molecules in a microscope. Like, why would they respond in the first place? Or what exactly is this nonlinear optical response? How can we understand that? And one way to do that is to visualize this molecule as, just a bunch of electrons that are confined on a molecule that are starting to move when the electric field of light comes in and starts to drive them. Okay, so you get basically an oscillatory motion of your electrons that move within the molecule. They move at the optical frequency of the light, of course. These electrons are bound to their molecule. They don't fly off, and they are bound by a harmonic potential. So that's indicated by this guy right here. And the nice thing about this harmonic potential is that if you plot the polarization that you create because of the shaking of that electron, moving the electron away from its equilibrium position, that the polarization scales linearly with the incoming electric field strength. Okay, so when I come in with my driving field, this is my light field here below, and you can see that the polarization is a function of the magnitude of the electric field. It's just a linear function, okay? Which means simply that the polarization just mimics the shape of the driving field oscillation, all right? So that's so far nothing really surprising, but surprising things happen if I'm changing the shape of this potential. Okay, I can imagine if I look at this potential that if I'm driving my electron now from an equilibrium position here at zero away towards the right, that is now more difficult because this potential is like going steeper up here on this side. It's more shallow on this side. So on this side, it's more difficult to drive than on the other side. So this is an asymmetry that I'm creating. And this asymmetry leads immediately to an asymmetry in the polarization that you create in your molecule. So, one reflection of that is that on this side, it's more difficult. So you need to move out the electron farther to get the same polarization. And on the other side, it's easier. So you get a pretty high polarization, negatively signed, but still much larger in size. So you get this kind of nonlinear dependence of the polarization on the electric field. And that leads to kind of distorted shapes of the oscillation. You can see here that now on one side, the negative side of the oscillation, you see it's much deeper, and on the positive side, it's kind of truncated. So this is now a distorted waveform. Okay, so you start distorting the waveform if you lose this linear dependence, right? So if you kind of take this distorted wave shape and then you decompose it into its Fourier components, of course, what you'll see, you get like overtones that contribute now. So the fundamental omega, let's say, fundamental frequency is there, but you also have overtones that contribute to the signal. And if you write it out, you see the polarization simply oscillates, you know, at all kinds of frequencies, right? And these are the frequencies that actually represent the radiating polarization that you can detect in your experiment. So all of these signals are related to the fact that you're shaking your electrons in kind of like an asymmetric way, you know, away from their equilibrium positions. If that happens, then you get a nonlinear signal. Now, that happens for all materials. Some people think you need special materials to do that. No, all materials have, to some degree, an optical nonlinearity. So this is a very universal principle. Optical nonlinearities, the only thing that you require is that you kind of drive the electrons hard enough so that you can sense, the electrons feel the non-symmetric shape of the potential, or the nonlinear shape of the potential. As soon as they feel that, you will see a nonlinear optical response. All right, so the simplest, the lowest order one is kind of like this 2 omega 1, okay? This is the second order response, third order response, fourth order response, and so forth, okay? So let's have a quick look at this second order response. And of course, that is responsible for second harmonic generation. Okay, now this is a picture you see in a lot of textbooks and in papers, but this picture is kind of like almost separate from what I just told you, okay? Because what I just told you is a classical explanation of optical nonlinearities, namely shaking of electrons and trying to see if there's like Fourier components in the motion of the electron. This picture you see here is actually a quantum mechanical picture, okay? It has levels, states of the molecule, and it also has arrows, and those are, of course, are the photons. So you see the energy of the photon in relation to the energetic states of the molecule here. So this lowest one is the ground state of the molecule. You see here another state called V, that is a vibrational level of the molecule. So these levels correspond to ground state vibrational modes of the molecule. And then you see a dotted line here. This dotted line means there's no real state of the molecule here, okay? No real state of the molecule. I just call it K, but this is called a virtual state. So what happens in second harmonic generation is that I have two photons that I stack on top of one another, and their combined energy is hitting something, but there's nothing there, okay? At least not in this particular case. And so it can't really park its energy here. And the result is, or one of the results is that the process is able to generate a photon with the combined energy of these two guys and bring the whole system back to the ground state. Okay, so this is a quantum mechanical picture of that process. So if you put input light of 800 nanometers, you can see that if you combine the energy of these two photons, of course, you get light out that's supposed to be 400 nanometers. So you kind of half the light, right? So the interesting thing about second harmonic generation is that the process responsible for this is subject to certain symmetry rules. I won't go into the details. Actually, if you're interested, you can read my book about it. There's a whole different But the outcome is that you need to have materials that have a certain kind of non-central symmetry, okay? And it turns out there are not really a lot of materials in biology that have that property on the length scale of the optical focus, okay? But people don't tell you that if you are very small, if you are a very tiny person, as big as a molecule, then everything else around you is basically non-centrosymmetric. There's very few molecules in biology that are actually purely centrosymmetric, okay? The problem is, if you are having a focal volume, which is about a micron or so, then you have a lot of molecules and you have all kinds of orientations. On average, this non-centrosymmetry, this centrosymmetry, it disappears, and you end up with a situation where, sorry, I say it the wrong way. The problem is if you have so many molecules contributing in your focal volume that this very specific case of centrosymmetry disappears due to averaging of orientations of all the molecules together, okay? So even though individual molecules, almost all molecules, are able to generate second harmonic generation if they are on their own. But if they are with a lot of peers and they have different kinds of orientations, the orientation actually factors into the phase of the light that comes out. And therefore, you get a phasing effect that averages out and depletes the signal if molecules assume all kinds of orientation. So on the length scale of your optical focus, there is really no second harmonic generation if you have kind of random orientations of your molecule, which is kind of the case in biology, all right? There's only a few structures where there is a persistent orientation that is sufficient enough to maintain the phasing of this effect, and therefore you counteract this destructive interference effect. And collagen type 1 is the primary example of this. It's just such a beautiful structure that does this on a length scale that is extended way beyond the size of the focal probing volume. Okay, and that's why you get images like that. Beautiful structures. You see these wavy patterns, very pretty. And the interesting thing is, again, this is not fluorescent. This is the second harmonic generation. If you look at this collagen type 1, there really is no absorbing quantity in there. Okay, you're just shaking electrons. And the fact is that it's so persistent across this entire structure. The phasing is just adding up, and you get a depleted signal in the end. Other than that, there's nothing special about collagen, okay? There's nothing special about collagen. It doesn't absorb more than most molecules. It has no particular features, molecularly speaking, that make it an outstanding structure. It's just their orientation on a longer scale that does the job. There's a few more structures that can do it. Microtubules have somewhat similar properties, okay? You can see also this is a persistent structure that remains kind of oriented in the same way on the length scale of the optical focus, and therefore it maintains some of its second harmonic generation capabilities. Myosin is another structure that can do it, okay? So all these three structures are great targets for second harmonic generation. And the nice thing is you don't need to label these structures now. You can just see them. You can see them specifically, okay? Nothing else around will contribute, but these structures will. Two-photon excitation, of course, I think most of you are familiar with this, very much related to second harmonic generation, but in fact here, there's a fat line here, which means this is the real state of the molecule that I'm exciting. Now I can park energy in there, okay? Part of that energy will relax and then fall back to the lowest emitting state from which I'm gonna harvest this particular photon, omega 2, which is a different wavelength, and I can simply just put a filter there and just detect that light, okay? So that's two-photon excited fluorescence. Here you see a graph where on this axis right here is a one-photon excitation spectrum. You can see here's a little maximum here, a little bump here, and then a big maximum over here. But if you take the two-photon excitation spectrum of the same molecule, you can see it here on top, okay? So this axis is basically twice the axis below because I need two photons to do it. You see it follows somewhat the features. So you see the bump here, okay? That corresponds to the bump over here, although it's shifted. Second bump vaguely responds to this, but this third one doesn't show up, okay? So it's not a one-to-one match with your, twice the energy of the absorption. It's not like that, okay? And the reason is that there's different selection rules, okay? So if you have two photons exciting one state, a particular state, you need to kind of abide by certain parity rules, okay? If you took a course of quantum mechanics, you may remember this. The parity rules for one photon excitation are different from the parity rules of two photon excitation, and therefore you are actually exciting different states. The interesting thing is that most of these states that you excite will fall back to the emitting state, okay? So the fluorescence is completely insensitive to the way you excited it, but the absorption is. So the excitation spectrum looks different, even though the fluorescence emission spectrum looks the same, okay? This is the two-photon quantum yield of certain chromophores, and it's indicated in Goupert-Myers, and these are typical kind of quantities. Again, for different chromophores here, you see a lot of interesting and useful chromophores have great kind of two-photon fluorescence quantum yields in the range, which is a good range to be at for two reasons. One, there's many laser light sources that are readily available to actually access those excitation wavelengths. And secondly, this is exactly in a window where scattering is low and absorption is low in tissues. So a great situation. So keep that number in mind. 100 GM, 100 Goupert-Meyers, is a really good number for two-photon excitation. So in tissues, there's several structures that have an intrinsic two-photon absorption that is very strong, on the order of 50 Goupert-Meyers, or maybe sometimes 100 Goupert-Meyers. Well, 100 is really high for endogenous, but any case, sufficient Goupert-Meyers to actually see the signal. This is elastin. You see it here in green. Okay, elastin, of course, is a fibrous protein, and it contributes to the rigidity in all kinds of tissues, elasticity in lots of tissues, but it also actually has a chromophore quality to it, and it emits. Okay, it has a pretty good fluorescent yield, and therefore you can use two-photon excited fluorescence to see it surprisingly efficiently. NADH is, of course, a much smaller metabolite. It's not a protein, but it is actually a pretty good molecule to address through two-photon excitation, and it allows you to see individual cells. Okay, you can do also metabolic studies with it, of course, but in tissue imaging, it is beautiful because most cells, of course, produce NADH in pretty large quantities, which allows you to see the interior of cells. The perinuclear region is usually pretty dense in NADH, but the nuclei is not, and therefore you see these wonderful dark holes, which immediately tell you what a nuclei are. Okay, so I don't need a nuclear stain here or anything. I can just see the cell morphology through the distribution of intracellular NADH. Okay, so you didn't have to do anything here. No special labeling protocols or whatever. You can just go into a person's arm and see the individual cells morphology completely laid out for you. Melanin, of course, is a very strong absorber. It's actually a pretty bad emitter, but interestingly, if you do two-photon excitation, you actually reach levels that are somewhat better coupled to the emitting state of melanin, and so therefore two-photon excited fluorescence in melanin is actually not that bad, even though one-photon excited fluorescence of melanin is pretty bad. Okay, this is one of the reasons why you as a person are not fluorescent in regular light. Okay, regular illumination conditions, you are not luminescent. But if I would put you in a two-photon excitation laser, you will be. Okay, much higher quantity of fluorescence will come out. So two-photon excitation fluorescence is a good way to study melanin. This is an example where you can use actually lifetime imaging to discriminate two different types of melanin, eumelanin and phenylanin. They have slightly different lifetimes, and that allows you to do some very important studies in melanoma. Okay, because phenylanin and eumelanin have different kind of malignancy qualities to them. This is one of my favorite pictures ever. It was taken by our colleague Mihaela Balu, and it is basically a recording of somebody's forearm using now two-photon excited techniques. One of them is second harmonic generation that we already talked about. The other is, of course, two-photon excited fluorescence. And this microscope has two detection channels. So one excitation, two detection channels. One of them is second harmonic generation. The second one, of course, is two-photon excited fluorescence. And what you see here is a function of depth is different structures contributing in different ways. Okay, so you see beautiful kind of demarcation here. Okay, this line here, which is the basal cell layer that separates the kind of the dermis from the epidermis. Okay, so the whole green structure here on top is all epidermis. And as soon as you see this blue color come in, that is the dermis below it. Just wonderful to see that in a living person. No labels, nothing. You see the individual cells, you see the nuclei beautifully stand out. You see the basal cells kind of like emanating from the junction, right? You see the papillar collagen right underneath through second harmonic generation. And then you see below that, the green structure here in the dermis, that is the elastin. So the fluorescence from the elastin, the second harmonic generation of collagen gives you all this fibrous structure information, right? Well, in the epidermis, you see all the cellular information through NADH fluorescence of the kind of the keratinocytes that basically constitute the epidermis.
Sorry, I think this is a very basic question, but why is the dermis blue?
The dermis is blue because that is where collagen is found. There's no collagen in your epidermis, and therefore the blue color is the second harmonic generation from papillar collagen in this case.
Yeah, if you go to the first one. Okay.
All right, so as beautiful as this picture is, I can tell you that the depth here from the top to the bottom is 120 microns or maybe 150 microns, which is enough to kind of capture the transition from the epidermis to the dermis, okay? At least in this part of the forearm. But it's not generally true. I mean, if you go to different parts of your body, then this epidermal layer can be significantly thicker, all right? And so you will not be able to reach the junction. And in many cases, you would like to really get into the dermal layer because that's where many pathologies actually originate, okay? And so it is actually really important to be able to move beyond a few hundred microns, okay? So this image, as beautiful as it is, it's kind of an exceptional case. So how do you actually get deeper? As you can see, the deepest locations here at the bottom of this picture become very fuzzy, okay? There's still a signal, but the features are blurry. Of course, the signal is weaker, but also it gets blurry, the contrast goes away, and that is something that we like to overcome. So if you are here at the top, this little spot right here is my focal spot that is kind of unaberrated, not affected by scattering yet, and therefore it's very spatially confined, producing very strong signals. But if I go deeper, of course, what happens due to scattering, I'll lose a lot of photons, so it's weaker. And secondly, it's distorted, so it's much bigger. So there are two effects that I don't want. So I want to kind of crank up the power. I can do that, and a lot of people do that. You just I'll lose photons, but if I just send more in, then in the end, I get the same amount where I want them to be, okay? And so then I collect the fluorescence from that spot regardless. But the problem is, if you do that, then you have to send more photons from the top here. And at the top, there can be so many photons going in that you start generating signal from the top as well, okay? So the flux is so high that two-photon excitation out of focus two-photon excitation actually starts to come up. And this is an important graph. This is from a paper by Chris Hsu from Cornell, where he looked at an important quantity, that is the fluorescence from the two-photon or three-photon process over the background that you create by any means, okay? So the signal over its background. And as you can see, in the magenta here is two-photon excitation of fluorescein in a brain, okay? So they basically did perfusion in blood vessels with fluorescein, and they see like at what depth am I losing the signal. So you see here the signal is pretty steady for a while. This is in millimeters, so 1 means 1 millimeter here. And so you can see you actually dip down pretty quick. This is a logarithmic scale once again, if you do two-photon excited fluorescence of the fluorescein probe, all right? Now, if you go to three-photon, then you see you get the same, more or less, under the conditions examined here, but it stays steady for quite a bit longer. And at this location, 1 millimeter, if you compare 1 millimeter here, of course you go down, but comparatively to fluorescein two-photon excitation, you can't even see anything, okay? So the three-photon excitation is kind of your only game in town at these depths. And that is directly related to the graph that I showed earlier on, where you saw that those tails, actually of three-photon excitation, come down way quicker, okay? And so the out-of-focus contributions are suppressed much more efficiently in three-photon than they are in two-photon. All right, so the last thing I'd like to bring up is vibrational spectroscopy methods in microscopy. I mentioned already two techniques that can do it, CARS and SRS, and they are based on the Raman response of molecules. And here you see a Raman spectrum of a very generic biological sample. Actually, this is a virus, okay? But the interesting thing is lots of biological samples have the same kind of basic structure in the vibrational spectrum, okay? So you see always a very big peak here on the high energy side. This is called wave numbers. This is of course reciprocal centimeter unit, but the unit itself is called, the quantity itself is called wave numbers. It is directly proportional to energy. So this is basically proportional to vibrational energy. The high energy vibrations here are the OH stretching vibrations of water. And the second largest peak are the CH stretches of organic compounds in your biological structure. And then you have nothing here for a long time. Okay, so there's really no response between, you know, this high energy range and then this other energy range where there's lots of little tiny peaks that tell a detailed story about which molecules contribute. So every peak here corresponds to a particular vibrational mode of particular molecules. Okay, so it's your task to figure out what's going on. But if you're good at this, then you can use it to your advantage and basically target specific molecules based on their specific vibrational fingerprints. That is a general idea. So the Raman process in the same kind of quantum mechanical picture can be visualized as follows. I have the ground state again, and then what I want to see is this vibrational mode, okay? This ground state vibrational state of the molecule. I want to excite that. And the Raman uses a very interesting way to do it. It actually doesn't do it directly, it does it indirectly. So you send a photon in, you don't excite a particular eigenstate of the molecule, but you let spontaneous emission happen such that you couple back to this vibrational state. So it's an indirect excitation process. Now, this indirect excitation process, as you guessed it right, is pretty darn inefficient. Okay, and the reason why it's inefficient is twofold. First of all, there's no state there. Okay, so the molecule is not very responsive. And secondly, this photon here is different from the photon here, different color. Where does it come from? Okay, that has to be spontaneously created. Okay, it turns out actually it's spontaneously created by quantum field fluctuations, but it is a very inefficient and improbable process. So this photon, the birth of this photon, omega S, the Stokes shifted photon, is a very inefficient process. So as great as Raman is, it is inefficient and is usually not really great for imaging purposes. And that's why people like to use, you know, what seemingly looks like more complicated stuff, but it turns out that in the end, you get more photons out, and that's all that matters. Okay, so the first step looks very similar, as you can see here. Okay, the first two arrows look very similar to the two arrows for the Raman. But then I'm having another step here, one arrow up and then one arrow down. And this process here is called coherent anti-Stokes Raman scattering, a 4-photon process. Okay, but the beautiful thing about this process is that, unlike the effect here in regular Raman, which happens independently for all molecules, okay, there's no particular phasing between them. Here, I'm forcing all those molecules to respond in concert. Okay, the reason is that this omega S photon here is not spontaneously emitted. It is actually a laser beam that's coming in. So omega P and omega S, pump and Stokes, they are both in the sample. They are together actively driving the molecule. All molecules are driven in the same way, in phase. And that means that with the last beam, this pro beam, omega P, I kind of scatter off of them in such a way that all photons that come out are in phase. So I have basically created a situation where I have coherent Raman. I have basically made sure that all molecules reinforce one another in terms of their emission, and therefore I get a much stronger signal. Seems like magic, but it actually really works. Okay, the difference between regular Raman and coherent Raman can be really dramatic, up to 6 orders of magnitude. And that is extremely significant if you want to do imaging. I mean, 6 orders of magnitude in terms of time spent in taking an image is just absolutely enormous, right? So that's why this trick is really useful in practice. So this is just to illustrate that. This is a picture, I didn't take it myself, one of my colleagues took it. Beautiful picture of a mouse brain. There's a huge tumor there, as you can see, okay? And what they did here, they mapped the whole thing out in two colors. One of the colors was indicative of lipids, and basically that's the green, and it indicates all the myelinated neurons, okay? And in blue, you see protein density, okay? And you see this big blue structure here. That means there's neurons, there's really are no neurons here, at least not myelinated ones. And there's a lot of protein, so that's really where the tumor sits. So this image is gigantic, lots of data points. You could never take this in a regular Raman microscope, but the nonlinear optical microscope, way faster, allows you to take this, again, no labels, but still chemical contrast, okay? I showed you this image in the beginning, and I said it's going to be important later on. Now we are later on, and so let's talk about it, because there's a phasing effect, and I just told you that this CARS process and all coherent processes depend on the phasing of these molecular oscillators. The way they shake and their position in focus is determining how the photon that they produce will come out and interfere with the photons produced by other molecules. So let's say I have now my focus, it's now horizontally oriented, and I have here, over there, this is my detector, okay? So I have just a hemispherical detector in the far field. The distance, of course, is a large distance r away from the focus. So this is not to scale by any means. But let's say I have a point A and a point B. They're both in the focal cigar. One of them is now radiating, and let's look at the radiation that goes all the way and is detected here on the detector at this particular spot, okay? Now, I also would like to detect a photon from B, okay? So both A and B will be producing a photon that I'll detect here in that spot. Now, you can see those arrows actually have different lengths, and therefore it may very well be that the length of those arrows corresponds to a phase shift between the two that leads to destructive interference. And that's typically what happens. The more you move away from the center position here to the outer edges away, so larger angles, that means that the chance is way higher that your two photons from two different positions will be out of phase. This is an effect that doesn't play any role in fluorescence microscopy, but it does play a role in coherent microscopy. So if I now do the following, I'm going to just walk across this hemispherical detector at all points and see what is the intensity that I can find if I add up all the molecules in this cigar that start to radiate, okay? And that's what you get when you plot that effect. You see this like lobe structure. So this is basically the intensity that you see as a function of you walking on that far field hemisurface. Okay, so if you are in the middle of a hemisurface, right there, then you are right there, okay? It means maximum signal. But if you walk away from it, so towards the edges, you see, it goes down, 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 till 0, which is like all the way 90 degrees, then you see nothing, okay? So this is kind of like the profile, the far field radiation profile that comes out from this coherent process. And that has really funny effects. So let's look at one particular spot, like one molecule. What if I do nonlinear optical microscopy in the coherent domain, and I just kind of drive that and look at the emission profile of a single molecule? You get this kind of donut shape, and most people recognize this as a Hertzian dipole, and that's because it is. It is actually just the radiation of a single point dipole, okay? That's what you get. But in microscopy, we never have typically one molecule that contributes to the signal. We get many, many more. So what if those molecules are oriented in this or structured in this kind of, film right here, this kind of thin film across focus? Then you see something like that, okay? So how do I interpret this? Well, I see there's signal going up. There's also signal going down, but there's no signal going towards the left and right. Okay, so the lateral contributions have been kind of destructively interfered out. Look at this peanut shape. That's the emission profile. If I extend it, I get even more peanutty. Okay, now what if I just fill up the entire focus with molecules that produce a coherent signal? Then, interestingly, what happens is that my peanut shape disappears, and I get nothing in this backward direction. Okay, so this is basically for negative angles. This is the backward direction. Everything is going to go up. So that is because all those, oscillators, they produce photons that only constructively interfere in the forward direction, and therefore radiation is very strong in the forward direction, but destructively interference is prominent in the backward direction. Okay, if I fill up only half of my focus, then still a lot of destructive interference in the backward direction, but there's still something left. Okay, just a little bit, as you can see here. So to make a long story short here, the emission profile and the amount of signal that you get is highly dependent on where the molecule is and how big the object is. Okay, and this is something that is not really playing a role so much in fluorescence microscopy. So this is a nice illustration of that. This is Carr's microscopy. And you see here a signal detected in the forward direction and a signal detected in the backward direction. This is a computation here on top. So in the forward direction, oops, I don't know what happens here. Forward direction, you see the dotted, oops, wow. In the forward direction, you see this line going up, 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 and it kind of saturates after a while. If I change the size of the object, so the bigger the object is, the signal grows and then it flattens out. Okay, that's what it says, the dotted line here. Why does it happen? Because I grow forward, everything is constructively interfering, very good. At some point stops. Why? Because the structure is now so big that it fills the entire cigar, and there's really nothing to gain because I don't drive molecules anymore. So that's why it flattens out. Okay, so you get this kind of plateauing. In the backward direction, it grows too, goes up, 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 up. Then maximum is achieved, and then it wiggles and goes down kind of gradually. And that's because it first grows, but then actually destructive interference is setting in. And then destructive interference is kind of like destroying it. And then you get this kind of oscillatory balance between constructive and destructive and constructive. Okay? And so eventually destructive wins, and you kind of deplete the so-called epi signal, the backward signal. If you don't believe that, then you should look at this image right here. This is the forward image of a cell, okay? You see everything contributes. That this is a CH stretch image. So basically, I see all molecules that are rich in CH bonds, okay? Carbon-hydrogen bonds, which is basically a lot of molecules. So basically, I just map out the cell. And what you see the nucleus here, you see some perinuclear region structure. But if I do the same imaging at the same time in the backward channel, and I see a very different image, okay? You see actually this membrane now is dark, and I see a lot of speckles and structure here that I don't really see clearly right there. And the reason is because the contrast is completely different. I have a size selective mechanism in the epichannel that I do not have in the forward channel. Okay, so the size selection mechanism suppresses everything that's big and enhances everything that's small. Okay, so a very interesting effect. Completely there because it's a coherent process. Okay, in incoherent fluorescence microscopy, you don't have this. If you don't understand this, then of course you have a hard time interpreting your images. So that's why these kind of simulations and this kind of modeling is important for us to understand what it is that we're looking at. Now, that sounds like bad news for us because, look, I am interested in backward detection because if I want to do tissue imaging, I can't really go through the entire tissue. If I look at a real animal, I cannot really go through the entire animal and collect the photos on the other end. I want to do backward detection, backscattered light. But according to this simulation, it looks like I got nothing. Okay? But that is, of course, not exactly the case here. This simulation tells me how many photons you generate in the first place that are intrinsically backward propagating, okay? Why is that important to realize? Because in this process, most photons will be constructively interfering in a forward direction, not in the backward direction. However, the forward propagating photons will be backscattered at some point, okay? So if you start collecting in the backward direction, what you basically are seeing is forward propagating light that is then redirected in the epi direction. And that is basically what you see when you do tissue imaging with this kind of cars or also second harmonic generation type of imaging. All the signal is initially propagating forward, but then is backscattered due to tissue structures. And that is what you are collecting on your detector. This is a live mouse. This is a video rate. Again, we're tuning to the C8 stretches. You see the sebaceous glands. This is a sebaceous gland. This is the hair coming out. See individual cells. The dark spots are the nuclei of those sebocytes. And so you can see with chemical specificity, particular cell types that are rich in lipids, because that's what this particular setting is sensitive to. You can do depth-dependent imaging, and you get this beautiful real-time recording of what's happening inside the skin of this mouse. Okay, so this is the first real kind of, video rate imaging demonstration of this kind of coherent imaging. People thought, it cannot be done. It's too exotic, but it turns out it's actually perfectly fine. And in some cases, this is really the only way to do it. This is another beautiful picture taken by one of my colleagues, Connor Evans from MGH. Again, this is also a sebaceous gland, but much higher resolution now. You see beautiful. You see this hair here, right here, right? That is the hair follicle. And you see this, ring, this horseshoe arrangement of sebocytes around it. Just an absolutely gorgeous image of the structure. And this then is the kind of a combination of different kinds of these techniques. You see here a piece of a kidney, and there's a blood vessel here. You see elastin here in green. Around it, you see the collagen, all right? You see a lot of other structures here. In red, you see the lipid through CARS, okay? So we have 3 different modalities, 2-photon excited fluorescence, second harmonic generation, and CARS. And together, they give this beautiful image. It's all label-free, but it's molecular specific. And that's kind of, very relevant in many of these studies where we'd like to pinpoint, like, what is it that we're looking at? Can I get extra contrast based on these structures? And this is the kind of contrast that makes, such kind of analyses possible. All right, I'm going to wrap up here. Some outstanding challenges. As I hammered on a few times, scattering is the main enemy of focus-based microscopy, especially in tissues. It attenuates and distorts, and those are not good things. And basically, in principle, it comes down to anything beyond a few hundred microns in most tissue types is just is out of reach, and it's a real limitation. Okay, so we would love to overcome that. And one of those things is that we need to better understand exactly what structures contribute to this scattering effect, and can we reduce it some way, somehow? We have to be smart about it. And can we increase the penetration depth, thereby, of course, improving image quality? So, I don't have the clear answers to all of this. Many smart people have thought about it, and I would say it's up to you guys probably to solve it. All right, thank you.
Question?
I'm back.
Any questions for Eric?
Now, who of you actually are doing nonlinear optical microscopy? Anybody? I see 2 hands.
Three hands.
3 hands, wow, okay. Now, what kind of techniques are you guys using?
I'm using 2-photon excitation fluorescence, and I do a lot of FLAM imaging.
Is penetration depth a limitation for you? Are you looking mostly at cell cultures or tissues?
I'm doing tissue. So yeah, penetration depth is very important for us.
Do you have a strategy for that?
I'm figuring that out right now.
All right, awesome.
At the early stages.
Yeah. Anybody else? Anything beyond 2-photon? Second harmonic, maybe? Everybody's using two photon. Two photon is great. I mean, it's just it's a wonderful technique. You need one laser. If you have the right laser and the right filters, it works. The signal is there, so it's a wonderful technique.
People are blown away, I think, Eric. Let's see.
That's right. I mean, I always put people on the spot, like, now it's time to ask questions. To validate the speaker.
There's a question.
There's a question.
Just sorry,
Right.
for Dr. Balou's image, what was the penetration depth there? You said it was a volar forearm, was that it? That was the
Yeah,
image?
it was a forearm. I think I should look at I should actually have listed that, of course, carefully. But I believe it was 150 microns. That was the extent of the entire image. So 150 microns is, it's not very deep, as you probably know.
Eric, can you give us a sense of the drop-off in the intensity of that image from the top surface to the bottom of that frame? Do you actually adaptively scan more at deeper layers than at the top? I imagine the intensities are quite different from the top down to the bottom. Or maybe am I mistaken?
Yeah, I mean, I have not done the analysis on the image. I mean, I think by eye, it looks like there's maybe a drop off by a factor of 2. But I also don't know exactly if she was giving a little bit more power flux at the bottom compared to, if she gradually changed that. I don't know. But, the bottom line is beyond 150 microns, it becomes very fuzzy, right? And in tissues like skin is one of the most challenging tissues, to be honest, because it's the refractive index differences are so dramatically changing within the first 100 microns that it's just really hard to combat. And so 150 is a very typical, even with those tricks. Even if you increase the power, you will never retrieve the resolution, because that's just, that's light scattering, which you don't turn off. And yeah, I'm not sure if that answers your question. I just started to talk.
So there are a couple of microscopy projects that involve developmental biology type research, and so I not being familiar with the field in the sense of the types of microscopy, does cars have a role in that sort of space?
Yeah, I think so. I mean, nowadays you don't have a cars microscope on every campus, but typically there's one around on a nearby campus. So there are commercial systems available. Leica sells one. There's many people in my community that have their own microscope that they work on with a lot of collaborators from their campuses. So it's not so exotic as it used to be. And there are many studies that are just focusing on just applications of this, right? And the emphasis has been for a long time and still is on lipid biology, because the signal is so strong. The Raman signal in coherent Raman of lipids is so strong. And the fact that you can differentiate between several lipid classes, in particular saturated, unsaturated, and cholesterol esters of neutral lipids, that is very important for a lot of pathologies, and therefore is of direct relevance for biomedical imaging. And so, yeah, there's a lot of work that happens in that space. And not as much, of course, as two-photon fluorescence, right? I mean, that is, I would say, ubiquitous. I mean, every campus has at least 3 of those systems. But I would say that it is a pretty large space in terms of its utility.
So Eric, what is limiting the dissemination and the propagation of these other techniques? Clearly they have very valuable and unique characteristics. Is it simply cost at this stage? Or do you imagine like 10 years from now or 20 years from now, whatever, I don't know, SRS or cars will be as ubiquitous as multiphoton?
Yeah, so a couple of years ago, I wrote kind of like a opinion article on this very topic. I think it was called Coherent Raman Microscopy, Capable Technique in Search of a Larger Audience. And you know, I stated, and I still believe that if a commercial system comes along that is affordable and easy to use, and I think many people would like to have such a system on their campus, in their facilities. And there are a couple of, I would say, commercial products that are starting to look like this could be it. There's a Leica, which has a cars microscope, and it can also do stimulated Raman scattering. It's very user friendly. It works great. People who have that machine love it, and it's heavily used. The main limitation are, the limitations are the costs and the fact that, you need some sort of maintenance. The cost is just too much. It's like $1 million, and that is typically not worth the price for many people. There are smaller companies now that have appeared in the last 5 years that make systems that are significantly cheaper. We're still talking about, maybe 400K, 300K. So, but that is definitely much more attractive because that compares well with regular two-photon microscopes, which are ballpark about, $500,000. So I think it's getting closer. I think those smaller companies probably will make a dent, and that will actually maybe bring down the price of Leica as well. Who knows? So I think the game has started, as far as I can tell.
Along that line, is the largest contributor to the cost the lasers?
Yes. Yeah, because basically the problem with coherent Raman is that you need, at least in the current incarnation, you need two colors that are kind of like separate, and one of them needs to be spectrally adjustable. And that is difficult to do with one box. So you basically you need you need one pump, and you need use that pump light to generate a second color, and that makes it intrinsically more expensive. So people are thinking about, can we make it more compact? Can we have different ways of generating the second color? Can we make a very broadband solution to this, so we have all the colors at the same time? So that is happening, but I would say that it's still not really where it's supposed to be from a I would say, utility point of view. I mean, in a academic lab, it works beautifully, but doesn't mean that it translates automatically to the general user.
So also, sorry, so is a low power car system an oxymoron?
Not necessarily. I don't think so. I think yeah, it's fine. I mean, I remember yeah, no, I will not bring up this anecdote, but I've seen a lot of technologies come down in price significantly. Just a matter of, if there's a market, then the price will come down.
Okay, I think we're going to be transitioning to tours, so obviously you won't be joining us, but I don't know if there's anything you want to say in terms of like a preview of the tour that they're about to have of your lab.
Oh yeah, so okay, yeah, it's too bad I cannot be with you guys, but my student has agreed to do the tour in her lab. So she's working on. I kind of alluded to it in the presentation. She has a setup that is able to do cars also in second harmonic generation, but the emphasis is on some frequency generation, and it's kind of like a sister of SHG, okay, second harmonic generation. So it's also sensitive to non-centrosomatic structures like collagen, but it has an advantage because it is vibrationally resonant. So I can tune into particular vibrational modes of the molecule, and I get the same images that look the same as in second harmonic generation, but they have chemical specific information in them, and that allows you to link their presence and morphology to structural information. So that can be very attractive if structural changes happen in your tissue, if things start to kind of like go out of whack, conformational changes and so forth. You won't see them on a molecular level in SHG microscopy, but you will see them in SFG because of this chemical specificity. So she will demonstrate, show you the setup. She's worked on it for 1.5 years and worked really hard on it. So hopefully you will enjoy that.
All right.
Uses infrared light, which is somewhat unusual. Mid-infrared light, not like 1,000 nanometers or so, but this is actually 3,000, 4,000, 6,000 nanometers. Okay, so really the mid-infrared range, which is typically not used a lot in microscopy, but as she will show you, actually it can be very beneficial in some of the studies to use that light.
Okay, thanks so much, Eric. Get better.